will be delivered by Dr. Bankim Subhash Chander. And the title of the talk is Assessment of Whole Head Magnetoencephalography during Transcranial Electric Entrainment of Brain Oscillators. Dr. Bankim received his BE in biotechnology from Dr. B.R. Ambedkar University, Agra, and moved to pursue his master's from IIT-Madras under the supervision of Professor V. Srinivasa Chakravarti. He received his PhD from the Department of Psychiatry at the University Hospital of Tübingen, Germany. His doctoral thesis focused on developing methods to assess magnetoencephalography datasets acquired during transcranial brain stimulation. After completing his PhD, he continued to work on electrophysiological assessment of chronic neuropathic pain patients treated with spinal cord stimulation at the University Department of Neurosurgery in Tübingen. He is currently working as a postdoc at the Department Behavioral Neurology hosted by the Leibniz Institute for Neurobiology, Magdeburg, Germany. His recent research focuses on investigating the role of spinal cord in processing of sensory stimulus. In addition to his scientific interests, he is a mindfulness trainer and plans to study neuroscience of mindfulness. Over to Dr. Bunkin for the talk.
Yeah, hi, thank you. Thank you for this nice introduction. So I'll just share the slides. Give me a second and we can start. Am I clearly audible?
Yes, sir. Yeah. You
All
are, sir.
right. Okay. Let's confirm if you can see my slides.
Yes, we can.
All right. So, I mean, you have already introduced the title of my talk. So, I will just give you a brief overview. I'll briefly start with the background of this talk, which will build up the basic information that is required in order to discuss the results that will show today. I will briefly introduce three research questions and address them one by one with a combination of 4 to 5 studies that I have done. Then I'll briefly summarize and then give a future outlook how we can use the results and how this can help in further advancing science and design new studies. And in the end, I would briefly acknowledge my peers and supervisors. Okay, so the general framework which I normally use for designing my studies is for try to associate some kind of brain activity with behavior. And for recording brain activity, I'm usually using in this specific study magnetoencephalography. Also, there are other studies where I'm using electroencephalography. And then while people are we do these recordings while people are performing some kind of behavioral task, and then use the perturbed brain activity which is related to the task and apply brain stimulation to modulate this brain activity, and then again measure what are the induced behavioral changes. And then this allows us to strengthen the link between, or maybe rather form a causal relationship between what is the ongoing brain activity and how it is modulating our behavior. So essentially, brain stimulation happens to be a tool for us that also helps us in understanding the scientific basis or the mechanisms of how brain oscillations are interacting among each other in order to give rise to a certain kind of behavior. So, introducing brain oscillations. So, I think most of you already know that. But brain oscillations are reflecting mainly synchronized activity of neuronal populations. So, you have a bunch of neurons that are firing in a synchronized way that can give rise to different kinds of brainwave patterns. And these are usually segregated into different frequency bands, as you can see on the right panel. And for this talk, we will be mainly focusing on theta band, which is ranging from 4 to 8 hertz, and alpha band, which is ranging from 8 to 12 hertz. So, and we know that these brain oscillations are associated with different brain functions. So, and you will see this later on in the slides. Uh, with magnetoencephalography, it has a high spatial resolution. It measures basically magnetic activity of the brain. So whenever you can imagine, whenever a neuron is firing, it creates an electric field, and the complementary magnetic field that is generated is actually picked up by magnetoencephalography. It has a very high temporal resolution. We can measure in order of milliseconds, and also this offers a larger spatial resolution compared to EEG, which has maybe up to 128 sensors, but with MEG it's possible to go up to 300 over 300 sensors. So when we are trying to estimate where the brain activity is originating from, we use beamforming as a method. So with different sensors, for example, if there is a source which is denoted as SR, and there are different sensors which are represented in as X1, X2, X3 in the figure, and this has a transfer function which is denoted by H1, H2, H3, and so on. And the whatever activity that is picked up is basically is a weighted sum of all these activities, and this is what is picked up by all the sensors. And now what we do, we use beamforming technique, which is basically estimating the inverse solution of what is picked up from the sensors to find to help us estimate where the activity of this of the is coming from. So and then we can also co-register it with an MRI scan to have a more anatomically precise location in the brain where the signal is originating from. And then we are using brain stimulation. So as you can see in this figure, you have a red electrode and a blue electrode. Usually the color coding is conventionally taken from the from the electrical circuit. So red is usually the positive electrode and blue is usually the negative electrode. And we can apply weak electrical currents through these electrodes, and then hope that there is certain interaction of these electrical currents with the ongoing brain activity that can introduce certain kind of behavioral modifications. So in this study we are using a different a new form of stimulation paradigm. Briefly explain you why we design a new paradigm. So, for example, let's begin with the first figure on the top, where I'd say 6 Hz TACS, which is transcranial alternating current stimulation. So, this kind of stimulation is usually applied at a constant frequency. Let's say, so the polarity of the current is changing between the set amplitude at a frequency of 6 Hz. And when we plot the spectrum of this signal, we see a large peak which is at the frequency of 6 hertz in the second picture on the right. So now the problem is, because the artifact of the stimulation signal is concentrated at the 6 hertz frequency, it is not really possible to see what's happening there physiologically. So it causes a large interference when from in the EEG signals and also in the MEG signals. And to overcome this problem, what we did, we actually used a high frequency carrier wave and modulated the amplitude of that wave at a lower frequency of interest. So in this case, the frequency of the carrier wave is at 220 hertz, and this was modulated at a lower frequency of 6 hertz. So the current signal is what you can see in the third figure, and at the bottom figure you see the power spectrum of this signal, and now you can see it's the artifacts are actually localized and concentrated in the frequency range around 220 hertz, which leaves the lower frequency around 6 hertz relatively free from artifacts. So using this technique, we are able to assess, and I'll show you how we can assess ongoing brain activity during the stimulation. Yeah. So the next question comes: is it really possible, even though this kind of stimulation signal the power is delivered at the higher frequencies? So is it really possible that the modulation frequencies of 6 hertz is also getting influenced by this kind of electrical stimulation signal in the brain? And this is what I'm going to uh, show you briefly in the coming slides. And now we also think about what would be the mechanism, because now the power of this signal is actually concentrated in the higher frequencies. So how is the lower frequencies getting having an interaction? And this is what I'm trying to show here is through entrainment. So for example, in the top figure A, you can see that the ongoing brain activity is probably represented in blue, and the applied stimulation signal is in the orange. And when you see at the beginning, the peaks and valleys of these two signals are not really aligned. But over a period of few seconds, what we observe is there is a phase alignment in the brain activity and the applied stimulation signal. And this is what we call as entrainment. And this is measured as phase locking value between these two signals, and it ranges usually between 0 and 1, where 1 represents absolute synchrony and 0 represents complete randomness in the signal. So essentially, when we apply brain stimulation, we are hoping that the phase locking value between the applied stimulation signal and the ongoing brain activity should increase as a reflection of entrainment. Okay, coming to the research question. So the first question would be about, is it possible to assess brain activity during this new form of amplitude modulated stimulation? And then the second question is if there is a functional interaction between the applied stimulation signal and ongoing brain activity. And then the third question is if it is possible to modulate behavior with it. So coming to question number one. So for this, let's look at the first signal of the first figure A on the top. So you see when there is no stimulation in the black curve, you do not see a clear peak. Whereas when you apply a stimulation signal, the power spectrum of the brain activity shows that there is a peak at 23 hertz. So this peak is essentially reflecting the artifact and not the ongoing brain activity. And then people were performing a voluntary button press task in this study, and what we you can see after the button press, around the range of after like 0.5 seconds to 1 second, there is increase in beta frequencies, which is in the range of 15 to 25 hertz. But when we do this during stimulation, the whole spectrum is kind of shifted, and it's impossible to somehow estimate how much is the increase in beta activity, which is actually physiological. In this case, the stimulation signal was applied at 23 hertz, and the algorithms that we use for reconstructing brain activity using beamforming fail with a conventional TSCS signal. So in this study, we did 4 volunteers who did this button press task, and we applied different stimulation frequencies at 1 hertz, 11 hertz, and 23 hertz over the motor cortex. And what we could show in panel A, you can see we can not only localize the activity originating from the motor cortex, but in panel B, you can also see that during stimulation of 11 hertz and 23 hertz, we are easily able to also recover. Also, in case of 1 hertz, which is not shown here, but uh, it's possible to recover ongoing brain activity. And unlike the previous graphs that I showed in one of the previous slides, um, the figures are more or less comparable to as if it's absence of stimulation. So this is the advantage which using amplitude modulation, so that now the physiological frequency of interest do not have this high concentrated artifact which is originating from the stimulation signal. Okay, so now the second question comes, like, is there a functional interaction with the brain activity? So to address this question, um, so maybe let me just go to the next slide, and then it would be easier for me to explain this. So we had a phantom a melon, and we had 6 healthy volunteers, and we were comparing the activity as in response to um, as in response to the stimulation of TACS, which is the conventional, and the new waveform, which is the amplitude modulated TACS. And the idea behind this is that human head has electrically active tissues, which represents basically neuronal membranes, which are basically lacking in a melon. So these are all plant cells, and they are not per se, in this case, as electrically excitable as neurons are. So the study design was simple. We had the first TACS, and then and the next block was of amplitude modulated TACS. And what we wanted to show that there is a differential response in the phase locking which is introduced. And coming to the results, so in case of melon phantom, you see that there is a sharp peak in the first curve on the top in the blue for a TACS signal. So this was actually at the 11 hertz, which is the stimulation frequency. But when we apply amplitude modulated stimulation, we see that the peak is actually not as clearly visible as it was in the case of regular TACS. For human head, again, there is a peak which is representing residual artifact at 11 hertz, whereas it's not so evident uh, in case of amplitude modulated TACS. And what we also find that the power ratio, which we measured at the modulation frequency to the carrier frequency. So in case there is some form of demodulation of this signal happening because of interaction with the tissue, then the power of the module at the modulation frequency as a ratio with the carrier frequency should increase, signifying that there is some form of demodulation. And this was significantly larger for a melon phantom. Sorry, for a human head compared to a melon phantom. So based on this, we had the first impression that there is some form of interaction which is happening with the brain, or at least the electrically excitable tissues in the brain, which are different from a from phantom experiment. And this was the first hints that we got that there is essentially some form of demodulation that is happening. However, the mechanisms are not are clearly understood, how the amplitude modulated signals are being demodulated, or at least not, if not completely, there can be partial demodulation, but the mechanisms are not clear to us as of now. Then we also look for phase synchronization, which is introduced by the amplitude modulated TACS and the conventional TACS. In case of a melon phantom, we saw more or less identical plots, which are showing an increase in phase locking at at 11 hertz frequency. Mind the difference, the scale of the human head is actually twice as that of the melon phantom, and we see a large increase in amplitude modulated TACS phase locking with the human head compared to the conventional TACS signal. So this was our second hint that the actual entrainment or the phase locking is quite strong in amplitude modulated signal compared to the conventional TACS signal. And this led us to the understanding that probably the amplitude modulation signal is a better tool for introducing phase locking compared to a conventional TSCS signal. Okay, so then we wanted to check if there is still there is a functional interaction further. So for this, we used a frequency tagging experiment, and so what we did. So basically, we used the phase and waves percept. Maybe just let me just skip to the phase and waves percept here. So in this figure here, you can see the phase and waves percept, and the phase is tagged with the noise of 15 hertz, and the waves part is tagged with the noise of 12 hertz. And what's known, in case of absence of any stimulation, People usually have an average time spent on each percept is around 4.2 seconds, as mentioned in this article by Parkonen. And what we did is basically applied amplitude modulated stimulation and modulated the amplitude at 12 hertz and 15 hertz. While the electrodes were attached on the occipital lobe, which is at the back, shaded in yellow. And what we found was basically an increase in the time duration spent on the phase percept for when 15 hertz stimulation was applied compared to when 12 hertz stimulation was applied. Also, this was equally valid when if we compare when the 15 hertz stimulation was applied to when no stimulation was applied. So based on these two studies, we could show that there is actually some form of functional interaction between the amplitude modulated signal of the stimulation with the ongoing brain activity. But then the next question is it really modulating behavior? So it's more of a cognitive task. To address this question, we used working memory as our construct of study. And what we did, we already know that the prefrontal cortex is involved in uh, performing a working memory task, and the oscillations are based mainly in the range of 4 to 8 hertz. So working memory is essentially reflecting temporary storage and processing of information. And to test this working memory, where we used an n-back task. So an n-back task is basically, if you so we presented participants a series of letters after every 2 seconds, and when they would see that the second previous matches with the currently displayed letter, then they have to respond that this was a match trial. And we were basically measuring response accuracy to assess their performance. And this is an example of a two-back task. So what? So what we were thinking that the original idea was that if we try to we can entrain these brain oscillations, we would be able to actually improve the performance. But there was also um, another contra hypothesis which was suggesting that if it's possible to maybe disrupt the oscillations, because the power is actually delivered in the higher frequencies and not at the in the theta range. The idea was like if entrainment can actually increase the power, but apparently that is not the case, like we have seen before. So then we proposed a contra hypothesis where we were saying that okay, it's probably going to disrupt the ongoing brain activity and leading to a worsening of performance. And this is exactly what we found. So in this study we had two groups. Group we received stimulation, and group B only received like a sham stimulation. They were told that they will be stimulated, but actually no stimulation was applied. The site of stimulation was selected as over the forehead, which is reflecting more or less above the medial prefrontal cortex. And their task was actually to have rest for 2 minutes and then perform their two back task for another roughly 2 minutes. And before the participants did the real study, they were familiarized with the task, and the data from this familiarization was used to actually find the each individual's theta peak frequency, which was selected for the stimulation in each of the for each of the individuals. Yeah, so we basically measured the difference in response accuracy between group A and group B for assessing behavior, and for assessing physiology, we were measuring mainly what is the change in power while they were performing the task, and also what is the change in introduced phase locking with the stimulation signal. So basically what we found that there was a decrease in or let's say there was an inhibition in increase of performance uh, of group A compared to group B. So basically the group A which received stimulation performed worse compared to group B, and because it was so the first session was the baseline which is without the stimulation, and then we have the intervention. When we took the difference from there is a small amount of performance improvement from the baseline session to the actual test session, but this improvement in performance was actually inhibited when stimulation was applied. In fact, they got a bit worse instead of improving. So in the regions which are shown here in blue are also the regions which actually show an inhibition in power increase which is required for performing the task, located in the medial prefrontal cortex region. So essentially, if while people are performing a task, we would expect an increase, and then we applied the stimulation. So instead of the expected increase, there was an inhibition and increase. So there is an increase, but it is not up to the mark for performing the task well. Then looking at the yeah, so again this is confirming the same results from the medial prefrontal cortex. And then looking at the results of phase locking value in these regions, we see a phase locking between the ongoing theta oscillations at each individual's theta peak frequency with the applied stimulation signal, which is also at the same frequency. And essentially these are again the same regions which are involved in working memory. As you have seen in the results of the power, there is increase in phase locking, which is signifying there is certain form of entrainment of these regions, which is basically interfering with performing of the task. Again, it's the same from the voxels that show an increase in power related to the task. But then we did some more additional analysis because it was not clear why this is happening. So we used a symbolic transfer entropy for this assessment, and the idea was that this is a non-parametric assessment of the signals based on probabilistic measures. So essentially, if you want to find out what is the information transfer between from region X to Y, you would need certain samples of the signal recorded from X and see how well you can predict the time series of X. And also you see you have the samples from the time series of Y, and then you predict what is the next time series of Y based on not only the past values of Y but also the past values of X. So based on this, we could actually see that when we measured information transfer while performing the two back tasks, there is a significant increase compared to the resting state. And And not surprisingly, when we look at the hippocampus and prefrontal cortex interaction, we saw that during the resting state, there is not much change in information transfer. But while participants received stimulation during the two pack task, there was a decrease or inhibition in information transfer during the task, which was not the case for the sham group. So based on this, we could actually say that there was certain kind of disruption between communication from hippocampus to prefrontal cortex, and possibly the theta oscillations that are seen in the prefrontal cortex are actually driven by hippocampus. So based on this, we could actually say that TSCS is actually interfering with working memory performance, and there's an inhibition in power increase which is relevant for performing the task. And then yeah, so this increase in PLV is always what we observe is under the stimulation electrode, but there have been other studies which have shown there is a remote stimulation effect as well. Okay, So to summarize from study one, we could actually show that an assessment of brain oscillations during the new form of stimulation is possible. And using this amplitude modulated stimulation, possibly we can interfere with ongoing brain activity by increasing or by showing entrainment, which as we have assessed with phase locking value. In the study 2, we could show that there is a higher phase locking value compared to TSCS. So probably uh, there is certain form of electrical excitable cells in form of neurons or glial cells, we don't know yet, but there is certain form of demodulation of this amplitude modulated signal which is happening in a human head and not in a phantom model. And beyond that, we also have shown that when you apply, there can be frequency tuned effects. So if the stimulation signal was applied at 15 hertz and the frequency tag percept at 15 hertz was given a preference, which was not the case in absence of stimulation. And thirdly, we can also target higher cognitive functions such as working memory and possibly entrain phase lock and entrain the prefrontal cortical oscillations to interfere with working memory. Okay, so coming to the future outlook, so in this case, we actually wanted to improve working memory, and we had certain ideas how we could do that. But before I go there, I think there are certain methodological things that can be improved, which is currently we are technically limited from the hardware side. So, for example, we can actually have auxiliary circuits which can be built into the recording hardware that can actually introduce a low pass filter. And this is actually going to improve how well we can see the physiological frequencies of interest. So usually there are low pass filters, but they do not have a very steep filter characteristics. So like for example, I know they're usually in the order um, of a third. I think it's a third order filter that is used. But I think if it's possible to improve these filter orders at the recording side, this might already improve how well we can record without having noise from the high frequency stimulation signal that we apply. And then for actually improving the performance, we can use something like brain state dependent stimulation, adaptive brain stimulation, or brain stimulation assisted neurofeedback training, which I will briefly give an overview now. So, in case of brain state dependent stimulation, we again identify a brain region involved in a working memory task, or this could be any task. But and then we estimate again the brain activity during this task, and by using the hardware, we can do some real time analysis of these signals to estimate the phase. And depending on when a certain phase of this ongoing brain activity is achieved, then we can actually initiate the stimulation. So for example, when we were doing the working memory trials, if we instead of applying the stimulation throughout the working memory task, but rather instead if we could have somewhat detected the phase of the working memory uh, during the working memory trial at the onset and apply the stimulation at the same phase, probably this would have helped in improving the communication between hippocampus and and the ongoing brain activity. This would which could have led to an improvement in working memory. Sorry. And then the next form of stimulation protocol is actually adaptive brain stimulation. So in this case, again, we are identifying a certain brain region, and then we estimate the brain activity during the task, estimate the phase, and update the stimulation protocol if we want to introduce some form of phase synchronization, or rather we want to or we do an anti-phasic stimulation that can change and this is something which we can always update the rule of what how we want to modulate the brain activity. So instead we can have certain trials where we are trying to facilitate the brain activity to perform the task better, where have we, in the other trials we can actually interfere with the ongoing brain activity. So essentially, this form of a stimulation paradigm certainly helps in forming a causal relationship between ongoing brain activity and how different ways of perturbing this brain activity can actually change their behavior. And lastly would be that we can use such kind of brain stimulation for neurofeedback training. So basically, again, we start the same way, like identify the brain region and then estimate brain activity during the task, estimate the power, and then display this power of these oscillations during brain stimulation, and and basically ask participants to train their brain activity during brain stimulation to get the desired effect. So for example, while performing a motor task, for example, you want to increase um, the alpha activity. So apply brain stimulation in alpha region and also give neurofeedback at the same time, and ask them to ask participants to further facilitate their ongoing alpha activity. Okay, so that brings me to the end. So first of all, I would like to thank my advisors. So Professor Surya Sokadar, Professor Christoph Brown, they have overseen this most of this brain stimulation work from conception to publication, whereas Professor Eschweiler was part of the review and he had given his inputs for the working memory tasks. And Dr. Stephen Robinson from NIMH, he was mainly involved in developing the algorithms that we have used for assessment of of brain activity during the stimulation, and Professor Niels B. Baumer, who has always given his feedback to how to improve the study, and always help with publishing of the data. And now we can take questions.
Thank you, Bankim. Any questions, anybody? So these are not a seminars, right? So people can ask questions. It's okay.
Definitely. I was thinking maybe people want to interrupt in between and ask,
Yeah.
but that's also possible.
Yeah, Pragati. Hi.
Yeah, hi Bankim.
Hi,
Good
Pragati.
afternoon. Hi sir. So
Hi.
when you were showing that working memory stuff, right, Bankim, you were showing some connectivity slides, is it? I couldn't follow that, so just wondering if you could repeat.
Let me go back to the slides. I think I did not do any connectivity analysis, so I'm not sure which slides you're
I mean,
pointing to.
information transfer, is that what you
Oh
mean?
yeah, the information transfer.
Transfer from, yeah, even I was curious about that.
Oh yeah,
Is
this
it
is
related to connectivity or what is? How did you compute the
Yeah.
information?
How did you find that information transfer?
so essentially. I didn't want to get into the mathematics of it. So there is a whole paper written by Stephen Robinson on calculating symbolic transfer entropy.
Is it like
So
Granger causality?
no, it's
Granger causality.
actually different. So we are actually using rank vectors for computing this. So essentially, the idea is coming from information theory, but we are not using the signal as it is. But we are actually for example, we use the bins of the data. So we have like 5 samples, and depending on how the samples are organized in this 5 sample bin, we give it a rank.
Okay.
So let's say uh, so we essentially get a number depending on what is the sample amplitude. So
Okay.
if you organize the sample amplitude, it could be 1, 2, 3, 4, 5. That's basically one rank, or it could be 5, 4, 3, 2, 1. That's another rank. So after computing these ranks in the signal, That's what we give the input into this metric. And the idea is, so based on the like we have continuous chunks of these 5 sample bins, one after the other, and the idea is to predict how a signal or these bins from signal X could predict based on the past of X and past of Y signal Y.
Okay.
That isn't that like it's like mutual information or transfer entropy, those kind of measures,
Yeah,
right?
exactly. This is the symbolic transfer entropy is basically the non-parametric version of transfer entropy.
Okay, okay. So that
Mhm.
basically then indicates your connectivity, right? If
Exactly.
okay. Oh, interesting.
And so with this, so we were using this from NIMH. They have developed this SAM beamforming toolbox, or they call it SAM suite. And this is a function which is already programmed in it, and we have extensively used this with our MEG data analysis. So for this specific analysis, you can actually assign different nodes over the brain depending on your region of interest. And what we did, we used this AAL atlas, and based on this atlas, we selected like I think around 90 points, which represents different parts of the brain.
Interesting. Also, one
And
more. Mhm.
yeah, and sorry, and I was just going to complete, and then we were computing information exchange between these different nodes. So, and there is a directionality involved. So
Mm.
you predict from X to Y based on X and Y, and vice versa. So, in this case, what I represented was basically sum of information transferred from hippocampus to prefrontal cortex and back from prefrontal cortex to hippocampus.
Okay.
So is it still directional then? Like because
No,
you're
so the results
computing.
which I'm showing here were not directional because we took average across the two.
Okay.
But we could have had a directionality also.
Okay.
Mhm.
Nice. And was there any basis on why you choose like 220 hertz, 6 hertz, and those kind of frequencies? Were they subject
Yes.
specific or?
Yeah,
Um,
for the working memory task, we had subject specific. So there was a familiarization session before actual experiment based on which we would actually compute what's each individual's theta peak frequency involved in the working
okay.
memory task, and which is also originating from the prefrontal cortex.
Okay.
And the previous data, this was more technically driven. So in those cases, we choose a fixed value, so 6 hertz, 11 hertz, 23 hertz, for example, in different studies. And 220 hertz, there was not really much of a. So the only idea we had that we want to have the stimulation frequency away from all physiological frequencies of interest. So the idea was maybe we are interested in up to 90 hertz of stimulation, up to 90 hertz of gamma band activity. So we were like, okay, let's double it and then push it a little bit out beyond that. So that's how we thought we'd do it 200, but then we went up to 220 because we needed at least 20 hertz of range. So when we have 220 hertz, then we can stimulate at 23 hertz. So 200 minus 23 would be, I think, 197. That would be the first peak that we would see in the amplitude modulated spectrum. And this is still outside the from the Nyquist point of view. We have like if you want to look up to 90 hertz, we want relatively free from artifact up to 180 hertz.
Very nice. Why did you say 20 over here, Bankim? Sorry. Like,
Yeah, theta is 6, right? How come this 23? How did it, where did it come from?
So 23 hertz was actually for the first study where we were using the beta button press and beta rebound.
Okay.
Oh, okay.
So, so when we were designing this, we were like, we will do a lot of experiments, but we don't want to. We want to choose a carrier frequency which we do not want to change. And so the idea was, even if we want to stimulate in beta frequency range, we should be able to do it.
Yeah. Very cutting edge, Bankim. Great work. Thanks a lot for sharing.
Thank you, Pragati. Thanks for your questions.
Hi, Bankim. Thanks a lot again. I have a
Yes, sir.
question, a very naive question. See, if
Sure.
theta is the main frequency in working memory, and if you stimulated theta, how come the performance went down? Should you
Yeah.
expect it to go up, right?
Yes, yes. So original idea was that we were expecting there should be an increase. So we thought that when we would apply 6 hertz stimulation, it would actually help in increasing. But eventually, when we did these studies with the phantom experiments, we realized that the power is not increasing when we are applying the amplitude modulated stimulation. And no doubt that's the case, because the stimulation, the powers of the stimulation signal is concentrated around the carrier frequency. And this was the main idea why we are able to analyze the lower frequencies of interest in the range of theta. So then the only thing that was left is how we can, so was the phase synchronization. And apparently that's not sufficient to increase the power. um, and finally, the mystery was actually resolved when we did the entropy analysis, this transfer entropy analysis. And that's when we found out that the communication between, so the transfer entropy between hippocampus and prefrontal cortex is actually decreasing during the stimulation.
Oh,
So that was the reason where, after which we were sure that why we see a decrease in working memory instead of an increase. So, so what we could actually conclude, but not really sure, that probably hippocampus is driving the prefrontal cortical oscillations. And when there is a phase locking with the applied stimulation signal, then what is happening is that the prefrontal cortex is not able to communicate or get synchronized with the hippocampus. So hippocampus is mainly suppressing like competing stimuli that are already encoded during the task. And if this theta communication is disrupted, then the competing stimuli are not able to get suppressed. And because of this suppression of these competing stimuli, the brain is kind of identifying, okay, this is a match or a non-match trial.
so I have a real basic questions. Thing is, if you want to increase power in theta band, why are you giving the AM signal? Why can't you directly give theta? You said something in the beginning, I didn't get it.
Oh, yes. So when we give the signal in theta, then we have a large stimulation artifact, and this is not allowing us to actually study what's happening during the stimulation. So
But
then
when
we
you give
have
AM,
to.
you are not able to increase theta energy, right? Theta power. So what is the point in giving?
So the point is that now with amplitude modulation, even though without increasing theta power, we can still interact with ongoing brain activity from the phase point of view. So we can modulate the phase.
Okay.
So previously, if we use a conventional TACS signal, then we are increasing the power and probably the phase also, but we cannot assess is what is happening during the stimulation. But with this form of stimulation, we can actually uh, see what's happening. We can actually make some assessment of the signals that are in the range of the physiological frequencies of interest.
Okay. Do you have any background papers on this? I would like to learn more about this whole protocol, like
So
review papers or something, and also your own papers.
yeah, I think since we were the first ones to do this,
Okay, okay, yeah.
so I think I can. So there is Witkowski et al. Also, I'm the third author on that publication. Uh, I will send you those publications. So
Okay.
this was basically. Let me share my slides again. I show you. Um, okay. How do I go back? A moment, I'm trying to go back in the slides. Is it possible to end the slides? Yeah. So I was talking about this slide mainly.
Yeah.
So this is where we actually show that how this method was developed for the first time.
Mm-hmm.
So here, if you see when there is no amplitude modulation, in the lower curve, you see there is a lot of noise in the stimulation frequency range, which is around 20 hertz,
Yeah.
23 hertz. And when we do amplitude modulated stimulation, now we can see, like in case of 10 hertz, 23 hertz, in the bottom rows, we are still able to see activity which is similar to what we see in case of absence of stimulation in the first row.
So when you give AM signal, you have increased power in to 23 also.
No, in case of AM, we do not see a power increase.
Then.
So this is why we had to also figure out if there is some form of interaction with the brain activity or not, because the first thing is, okay, we can apply brain stimulation and we can recover brain activity, but if there is no change in brain activity, how do we know if there is some form of interaction with the brain activity or not? So this stimulation per se can just do nothing.
Yeah.
So then we went ahead, did further characterizations where we calculated phase locking, and with human head, we saw that there was an increase in phase locking with the amplitude modulated stimulation, which is actually much larger than what we see in a conventional TSCS signal. And in case of a melon phantom, they were more or less at the same amplitude. So also I mentioned about the frequency tagging experiment, and we could show actually that during the amplitude modulated stimulation, people were able to give preference to the phase percept when 15 hertz amplitude modulated stimulation was applied. So based on these two things, we could actually show that there is certain form of uh, physiological interaction which is going on through entrainment.
Mm-hmm.
And then we finally did the working memory study where we could show that this was much more complicated actually, because it was not how we were expecting results to go. So we could actually show that there is increase in phase locking, and which is inhibiting increase in power which is required for doing the task. Instead of so in this while performing a working memory task, there is an increase in power that normally is expected to happen, but during the stimulation this increase in power is actually inhibited. So when we take the difference, this is actually showing that there is a decrease in power increase. Yeah. So this is kind of the complicated part, which took us a long time to resolve, and eventually we could solve this when we looked at the transfer entropy between hippocampus and medial prefrontal cortex. So if you look at the resting state, then there is so you take the difference between pre and post condition. They are more or less the same between the two groups, but when we are doing the top working memory task, then there is a drop in information transfer during the stimulation.
So this choice of carrier frequency, is it important? The exact
uh,
value, or is it flexible?
I mean, in this case, we have not studied the much. So we always had the same carrier frequency.
220
However,
hertz.
there is another paper by Neil Grossman. They
Mm.
are talking about temporal interference. So they have a stimulation signal of 1000 hertz and 1010 hertz, and
Wow.
then they say that we can do non-invasive deep brain stimulation, because when you apply these two stimulation signals on the brain, wherever they are interfering at that
Mm.
specific location, because of interference, it generates a beat frequency of 10 hertz.
Mm.
and from electrophysiology they could record a response to this 10 hertz firing pattern to this kind of stimulation in rats.
So that's alpha, right?
Yes, this is alpha, but
So the
we'll just
overall alpha goes up because of that.
so they show a raster plot for firing patterns based on
Uh,
which they said there was increase. So this was electrophysiology.
hmm.
I am not very sure if this they measure alpha power,
Okay.
but definitely there was 10 hertz of firing. The firing rate was actually 10 hertz as they
Oh,
computed from the raster plots from electrophysiological recordings. So this was directly like intracellular recordings.
so this makes brainstem very complicated with
I
all
think
these
so.
interferences between frequencies.
Yeah.
Wow. Okay.
So this is also a new form of stimulation which has recently come up.
Hmm.
So yeah. So temporal interference is still new and still difficult because in this case they are still trying to figure out how they can let's say focus how they can focus this, because if in case of temporal interference there are two signals which are interfering in a space. So and depending on where, so if the exactly in the midpoint, let's say spatial midpoint of the two interfering signals, you have an absolute modulation, but it also tapers out a little bit into the surrounding. So depending on the physiology, uh, there can be it becomes difficult to study because the focality is not precise in this case.
Okay.
So with at least with this method, we can still figure out with amplitude modulation where exactly the phase locking is happening, and probably that's the region where there is maximum focality of the stimulation. With temporal interference, it makes it a little bit more challenging, but nevertheless, we did try that, and I think because lack of hardware filters, it was a little bit difficult for us to have an estimate what's going on.
Okay.
This was one of the hardware's thing that I wanted to mention, that if we have hardware filters which are in the just above the physiological frequencies of interest, then we can still have a better estimate what's going on. But right now, this is purely algorithm and post processing.
Okay. Sounds good, man. Very exciting.
Yeah.
Anybody
Thank you.
else? Any questions?
Bankim, I just have one follow up
Sure, brother.
on the transfer entropy stuff. So
Yeah,
transfer entropy, as you pointed out, it's a very nice measure for
mhm.
directional information transfer.
Yeah.
But was there any reason why you averaged that out and really took out the direction for computing the information transfer between hippocampus and medial prefrontal?
Not really. I mean, this was like the easy, just for ease of presenting. Otherwise, it would have been too many plots.
Okay.
But the main message still remains the same. So the
Okay.
analysis was eventually done in order to make sure that there is only one plot for each of the claims that we want to make.
Mm-hmm. So it's essentially saying both hippocampus to prefrontal cortex as well
Mhm.
as the other direction is equally important,
Uh,
equally.
not exactly
Um,
No.
equally, not exactly equally. There are certain differences, I would say, but I don't actually remember now what was the difference.
So,
But
for example, if there is a delay, if it is
yeah,
plus tau in one direction, it will be minus tau in the other direction.
exactly. But
Something like that. Yeah.
but uh, but the network is more complicated. It's what we are. There are also other regions in the brain where which are showing changes in information transfer due to the stimulation. So based on
Okay.
the prediction, it's not exactly plus minus. There is also a certain variance in that.
Okay. So I
because
was just
we do it trial by trial analysis and then average it out.
okay. Okay, cool. So the average is really not disturbing any of the single effects. Then is the meaning, right? Like looks like the average is not diminishing the activity information transfer measure or something like I'm just trying to understand like the stimulation it's not really working only on one direction. It looks like it has its effect on both, and you're trying to take the average of both the directions to
Yes,
yeah,
exactly.
okay.
Exactly. And what I did not show, that there are other brain regions which are also showing changes in information transfer. For example, with Purkinje's, which actually we did not know how to interpret.
Mm.
And the Professor Eschweiler was mentioning that these Purkinje's regions. This is the region where the earliest signs of Lewy body, not Lewy body, these tau proteins or amyloid plaques, exactly, that can be detected in case of Alzheimer models.
Wow. I was just thinking it's mostly a visual percept region or something. It's
Yeah,
interesting that.
it's kind of in between. It's like precuneus is between motor and it's in the parietal cortex between motor and occipital lobes. But yeah, and we saw that hippocampus had the maximum um, reduction in information exchange with precuneus, and we were not able to actually um, figure out why that's happening.
Okay. Hey, if that's okay, I have one more question.
Go ahead.
Sorry,
Go
sir.
ahead, go ahead.
So, is there a reason why you chose the I think the cathode was posterior and the anode was in the frontal, right? Like you had a direction of current flow.
Oh, but let me tell you about the montage. We had one electrode position in on the forehead, which is kind of close to the medial prefrontal cortex, and the other electrode was placed on the parietal cortex, in the over the medial parietal cortex. And in case of alternating current, I mean, conventionally there is a directionality, but if the current is alternating, I think we cannot say it's directional anymore.
Okay.
Yeah,
Okay, interesting.
I mean,
Okay.
just because the device has a positive and a negative, but in case of an alternating current stimulation, this positive and negative doesn't really have doesn't have a meaning anymore.
I see.
Mhm.
Okay. Thanks, Bankim.
Sure, Prakriti.
Okay,
Thanks
I
a lot.
think Bankim, uh, we should call it a day.
Thank you, sir.
Right.
Thank you so much
Thanks a
for this
lot. But
opportunity.
let us explore this from a modeling point of view because this looks very fascinating.
Yeah.
Because
I.
now
Mhm.
how to choose your protocol and modulation is not
Mhm.
obvious, right? It's very like a very subtle thing.
Definitely,
So,
and yeah, I think we need actually a lot more modeling insights into how neurons would be responding. So I remember, like from the courses that I did with you, when we apply a negative current to an Hodgkin-Huxley neuron model, it still starts firing.
yeah.
But this is challenging the conventional wisdom. If you apply cathodal stimulation, which is negative stimulation, it is kind of suppressing
Mm,
firing.
mm.
But it's only a matter of how high is the amplitude of cathodal stimulation. It can still turn out to have exciting or increase the excitability. So
So yeah, let us catch up with
yeah.
Deepan. I don't think he's here today.
Mhm.
Let us meet with Deepan and see with any of the oscillator models. If
Oh
you can
yes.
explain, give a rationale to why this kind of amplitude modulation stimulation,
Mhm.
right, is giving the kind of effects you are seeing.
Yeah, that would be interesting.
It's like a
I
very
mean,
deep question. This
yeah.
thing.
I mean, most of the community working on brain stimulation have a very linear thinking coming from like basic medical physiology,
Hmm.
and I think uh, we know there is not of non-linearity in the
Yeah,
system at
yeah, yeah,
multiple levels,
yeah.
which I think we cannot explore this with the tools we have unless
Right, right.
it's
Yeah,
computationally driven with
yeah,
modeling.
yeah.
Mhm.
Okay, thank you guys. Thank you, Bankim, once again.
Thank you, sir.
So thank you. Yeah. We'll meet. Thank you. Bye.
Mhm. Bye.